Yeah, I was silenced. So again, good morning and welcome once again to our Lab talk of the innovation series. We appreciate your assistance and interest. My name is Aletia Cruz, product manager for microbiology at Lab. So let's start with the presentation since it is a bit extensive, and we will see that we're going to touch on several points to arrive at what the name is, combined use of traditional techniques and qPCR for the analysis of Legionella. So let's begin. What are we going to see? The points to review in this occasion, which revolve around Legionella, is first a quick look at the traditional method, that is microbiology culture, of course, versus the alternative methods available on the market. We will delve into one of these alternative methods. The most used is qPCR for the detection of pathogens, to give way to solution that we present today, which is Contagion Legionella. And then in closing, how a combined culture method and Contagion strategy can be adapted for a more efficient workflow in the laboratory. And finally, we will review all the products that we have available in Conda, as well as the available resources. Let's go on with the first part of the presentation to put ourselves in context about our microorganism of interest today, which is Legionella. Legionella is, as we know, an aerobic gram-negative bacteria that we can find in aquatic environments, of course, and basically in any type of freshwater environment. They predominate in this type of source, although it has also been identified in potting soil. It is an intracellular parasite of Protozoa, which helps Legionella to survive in virtually any environment. The ideal conditions for its multiplication or growth are in a temperature range of 20 to 55 degrees Celsius, the optimum being 35. However, as we can see, we have at the lowest one being 20, which is very relevant for a common form of propagation, that is air conditioning systems. Legionella also has more than 60 confirmed species and 70 different serotypes, and one of these species, Legionella pneumophila, is one of the most relevant at the clinical level. Now we will see why, and this species alone has 15 serogroups. So it is important to mention that Legionella can survive thanks to biofilms, since as in other bacteria, these structures, of course, provide shelter and nutrients, but this needs to be investigated further. On the other hand, as I mentioned a moment ago, Legionella pneumophila, especially the serotype 1, is the main cause of the disease we know as legionellosis, which may well present clinically as pneumonia, that is Legionnaires' disease, or Pontiac fever, which is like a milder form and more similar to the flu. In fact, during the beginning of the pandemic and before the rapid tests, Legionella and Enterococcus tests were carried out to rule out COVID, since the most common of these is pneumonia. Although, of course, there are other pathogenic Legionella species. The route of transmission, being a respiratory disease, is through aerosols. And according to the ECDC, in the European Union, an upward trend in reported cases was observed for this disease since 2018, which is why periodic controls and other control measures in water systems continue. Unfortunately, Spain is among the countries, or fortunately as well, Spain is among the countries with the most reported cases. So we have a good control on Legionella. In microbiology, when we talk about traditional techniques, it is known that culture media have been the standard for decades. And for this technique, And this technique continues to dominate the sector. And anyone who has experience with Legionella in the laboratory, either theoretically or in practice, will note that the ISO 11731 from 2017 method is complex and laborious, as there is a multitude of possible variations from the matrix with which to make a decision to follow one or another step, which will depend exclusively on the nature of the sample. And this first step is extremely important because Legionella is also a slow-growing bacteria, and the existence of interfering flora can affect it even more or even prevent it. There are options of thermal pretreatment, acid or non, filtration, elution, and 5 agars to choose, such as BCYE, GBPC, MWY, BCYE with antibiotics, and BCYE without cysteine for the confirmation, which of course is mandatory. The incubation times are long, reaching 10 days for isolation and 5 days for confirmation at least. We have here on the bottom on the right a picture of a colony of Legionella and of Legionella pneumophila on a BCYE agar plate, and we can observe it. It is bluish in color and glassy in appearance. So for more information about the ISO method for the analysis of Legionella, and I recommend a webinar we have already done, and it is uploaded on our website. In the Condalabs talk section, you can access the recording. In this section, we have, of course, many of other webinars from different pillars of microbiology, food, water, cosmetics, pharma, also some about molecular biology. So we invite you and encourage you to take a look at this section of our website, because you will surely find more than one webinar that is of interest to you. So continuing with Legionella, as we have seen, the culture method has several problems due to its complexity and high variability in dilutions, pretreatments, and culture media. We also find ourselves with a workflow from reception of the sample to results that is greater than 15 days, and this leads, of course, to delays in response and low productivity. 5 different media that are complex to prepare, which again affects time and resources, mandatory confirmation that lengthens the obtaining of results, and an important part that sometimes is forgotten, inclusivity, since some Legionella cannot be cultivated in this culture media. So in other words, we have delays, variability, lack of differentiation between pathogenic and non-pathogenic strains, viable cells that are not cultivable, and other problems that we encounter with the traditional method. And of course, this has become an important need to be resolved in the sector, especially due to the issue of speed in the returning of the results. We have several techniques available on the market, as we can see. And in this table, there is a comparison. And on one hand, we have culture media. Of course, it can include chromogenic media, ELISA as an immunological method, and real-time PCR or qPCR. Each of these techniques provides, of course, with different parameters of specificity, sensitivity, and speed to give positive and negative results. Also, there are different degrees of influence from both the matrix and the accompanying flora, and something very important, the experience necessary from the user to carry out one of these techniques. All of these offer interesting advantages, but also important drawbacks depending on the technique. And observing this small, quick comparison, we can clearly see the reason why qPCR is the main alternative technique used, since its advantages are numerous, even compared to those of other rapid methods that are not culture media. So let's enter fully into the world of qPCR and its use in the detection of pathogens. Again, it is impossible to talk about qPCR without first mentioning the PCR, which of course, the vast majority of us know or have heard of. The acronym PCR is the name of the technique, polymerase chain reaction, with which it is possible to obtain copies of a single DNA fragment present in a sample through amplification cycles. These cycles have several temperatures related to these stages, the steps of the process: denaturation, where the DNA strands are separated; annealing, where the primers bind to the target DNA; and then ligation, where the polymerases are incorporated using the primers and synthesize the chain of complementary DNA. The main difference between a PCR and a qPCR is that in the latter, there is a fluorescent label that allows the reaction to be quantified as the PCR reaction progresses. And for this reason, it is also known as real-time PCR or qPCR for quantification. There are many techniques for this, but one of them is the designing of DNA probes, that is, fragments complementary to the target DNA region to be amplified. This labeling consists of a DNA probe with a reporter, which emits fluorescence, and a quencher, which turns it off. At first, the quencher prevents the emission of the first of the reporter, but with the action of the polymerase, the quencher will be released, which then will emit the fluorescence that can be detected by the thermocycler. But of course, it must be a device capable of doing so, and with which the specificity is higher, since, for example, another technique are dyes, such as cybergreen. They're a bit common, unheard of, which is unfortunately non-specific, and it simply binds to double-stranded DNA, and therefore running the risk of increasing this amount of fluorescence emitted. And although it is faster, the staining, the dyes, it is much less specific. In this image on the left, on the bottom left, we see what would be a reading of a qPCR curve, and each color representing different bacterial species, for example. In this next slide, we have at a glance the difference between traditional and alternative methods. So we generally have a traditional method with a series of steps that can start with a deletion, enrichment, or concentration of the sample, followed by a second enrichment or treatment, then a selective isolation to arrive at the confirmation and results after sometimes, and of course, if we are lucky, a week of work. While in theory, with a qPCR method, we could obtain the results of the same sample in just a couple of days. So let's see how this would work when implemented specifically for Legionella. We have a method, again, ISO 11731, which begins with the pretreatments, dilutions, and concentrations. A membrane filtration, specific isolation for which we have to select the appropriate media, and finally the mandatory confirmation, with which on day 20 we would have hopefully our result. On the other hand, with a qPCR method, which in this case would be our Condagene Legionella kit, very importantly, according to ISO 12869, which we will see in greater detail later, we would start with the concentration and pretreatment of the sample on the first day, and on the second day we would do the PCR itself, both the DNA extraction and the detection of Legionella, and that same day we would have the results. So in total, we are talking about 2 days of work versus the 20 days required for a microbiological culture. So it is obvious this shows the main and best advantage, which is, of course, the improvement of time to results, the speed of this method, and there is no doubt. But is qPCR only about speed? So qPCR is a technique that provides much more than that. Quick results. However, it is true that depending on the procedure, most of them can be shortened. Most of the procedures can be shortened to just a couple of days of work. The use of probes, which is the case with Condagene, means that the negative results do not require confirmation, and therefore samples can be released without having to wait for this last mandatory step in the culture technique. It provides great sensitivity and specificity, since only a small amount is required, in theory only 1 copy of the target DNA, to detect the pathogen. And as it is specific for this DNA, the interfering flora does not affect it in any way. Also, the experience of the staff is not as relevant as when doing good isolation or streaking, since depending on the kit, the protocols are usually simpler than the traditional microbiology and do not require extensive manipulation or even a large number of reagents. Another advantage is that qPCR allows automation for laboratories that work with large volumes of samples to be processed every day. But of course, not everything is perfect, and this is the case as well. And however close qPCR may be, it also has its limitations, and I think it's very interesting and also important to know about them so we can fully understand the qPCR method. A PCR detects cells present, but not also the not viable ones, because these dead or non-viable cells will also be identified, since this technique will always recognize the target DNA regardless of the state of the cell, which would not happen, for example, in culture media. In some cases, the reagents may be subjected to cold transportation to ensure their stability, which makes the method more expensive. And not only that, but a breach in the cold chain could harm the product and its function. There is also the possibility of false positives due to the cross-contamination of amplicons from previous PCRs. And of course, this would reduce the robustness of the results obtained. And although quantification is possible with qPCR, there is not yet a one-to-one relationship between genomic units and colony-forming units. So to provide a solution to most of these limitations that we have seen of the technique of qPCR, we are going to present Condagene, which will merge all the advantages of qPCR, and at the same time will lessen some of these inherent disadvantages of the technique. The first thing we can find with Condagene is a series of controls per ISO 22174, which is a standard for the use of PCR as a tool for the detection of pathogens for both human and animal consumption. The use of negative and positive controls is indicated in this standard. Also, according to this ISO 22174, and thanks to the use of specific probes, confirmation of the result is not required. However, when positive cultures have obtained, it is recommended to confirm by microbiological culture. And additionally, Condagene also has included an internal control that offers a protection against the false negatives. That is, it is a way to ensure that the reaction is occurring correctly, since otherwise something would be inhibiting the reaction and a series of measures should be taken to remedy it. Another security measure provided by Condagene is the addition of the UNG control, which eliminates the remains of the previous amplicons. That is the limitation due to cross-contamination that we saw just a moment ago. And through the degradation, this is achieved through the degradation of the uracil bonds, the nucleotide using the amplification of the PCR, and that is not found naturally in samples, and thus its amplification is avoided. Also, Condagene is compatible as well with the viability PCR. It can be achieved by adding a specific reagent that would bind to free DNA before extraction and thus block its amplification. Therefore, only viable cells would be amplified. And one of the pillars of Condagene is that the kits are based on public and standardized methods such as ISO, DIN, or the FDA. These last ones are Germany and American standardized methods. And specifically for Legionella, we would be talking about ISO 12869 that we have mentioned before, if you recall, for the detection of Legionella by qPCR. In principle, this would avoid the need for accreditation. It would only be necessary as long as the company that implements the method is accredited by ISO 17025. And if the entity is not accredited, this would not be necessary, just a simple verification of the method. These products are heavily supported, as you can see, by standardized methods and bibliographic references. Contagion kits have very simple and detailed protocols to ensure a correct and minimal handling by the user, and its reagents have a high stability even in liquid solutions, so their transport at room temperature is possible without it causing problems in the kit's performance. We have carried out studies that demonstrate correct operation even after 2 weeks at 37 degrees Celsius. And finally, Contagion is a product that is highly compatible with thermal cyclers on the market, and thanks to the homogeneous number of cycles common to all the kits, the different Contagion kits can be used on the same plate, of course, different walls, that is performing a kind of multiplex. And since we have reviewed the solutions that Contagion provides us, now let's see the method for Legionella. We would start with the first part, that is the extraction of the DNA from the sample, a critical step to free the genetic material found inside the Legionella cells. For this, we have 2 extraction kits, both based on a lysis buffer, whose objective is to break the sample cells to release the DNA, which would be our target DNA, and that will then be amplified in the PCR. Both kits are optimized for their use with contagion detection kits, in which the influence of matrices or inhibition is very low. Validation also has been performed on complex samples with and without pro-sample heat treatments. And we have first the contagion complex extraction kit that we would use to obtain effective lysis if you have a sample with a high cell load or, for example, inhibition problems. It is composed of 2 different reagents, lysis buffer and the buffer. And then the second is the contagion quick extraction kit. As its name indicates, it is a rapid quick kit where the extraction is carried out in a single step with a single reagent, and that we have successfully tested with a wide variety of matrices. Both extraction kits work perfectly for gram-positive and gram-negative bacteria, so either of them would work flawlessly for our Legionella samples. With any of them, we can obtain a correct extraction of the target DNA to use in the next step, which would be the detection of Legionella in this case. The Contagion Legionella kit is indicated for the detection of Legionella in water samples by qPCR according to ISO 12869. As we have seen previously, it is a highly sensitive and selective product thanks to the use of probes that are specific for Legionella through a simple protocol and easy to handle. This kit consists of 4 different reagents, and in general, like all contagion kits, the color classification of the reagents is the same to facilitate its use and for the user to be familiar with. This first tube with the blue cap contains the probes and the primers. Then the green cap would correspond to the master mix, the ready-to-use mixture of controls, polymerase, nucleotides. Then the positive control with a yellow cap, and finally 2 tubes with a white cap that would be the PCR grade water. The detection kit is for 100 reactions, and the positive control could be used for between 20F and 50 runs. We have seen a bit of the theoretical part of what Contagion is, how it works, the advantages it brings us, and the products for Legionella. We will now review the protocols and what a real workflow would look like for detecting Legionella in our sample with Contagion. So To begin, we must first of all prepare certain reagents included in the Legionella detection kit. We would have to reconstitute the positive control, which is just simply adding 100 microliters of PCR grade water to the control tube, mix it well with the pipette to homogenize, and voila, there's the positive control ready to be used. Then on the other hand, we have to reconstitute the tube with the probes and the primers. And for that, we would add the entire content, 440 microliters of the tube with the master mix, which is the green cap to the blue cap tube. Again, we mix it very well. We discard the tube of the master mix that will now be empty, and we have our mixture for the qPCR ready in the tube with the blue lid. We recommend allocating both reagents into tubes depending on the needs of each laboratory, since once ready to use, both reagents must be kept frozen at -20 degrees Celsius. To speed up the work in the lab, of course, make it easier, more comfortable, but also because there are limited freeze-thaw cycles of these reagents, and if exceeded, their correct functioning would be compromised. We have done stability studies on this. Of course, before each use, we recommend to give them a spin once they are completely thawed. We continue with what would be the detection of Legionella by qPCR using Condagene. The first part that we mentioned is extraction and then detection. So for the extraction, we have a step-by-step protocol for each of the Condagene extraction kits. On the left, we have the complex kit, and on the right, the quick kit. In both, the indications of ISO 11731 for the treatment of the sample must be followed. With the treated sample, extraction would be performed and the filtration elution, of course. For the complex kit, we would have 1 milliliter of dilution. We would centrifuge for 10 minutes, 13,000 RPM, with which we would obtain a pellet. And then the supernatant is discarded. This pellet is resuspended with 0.2 milliliters of reagent 1. We take the suspension to the thermoblock and program for 5 minutes, 1000 RPM at 85 degrees Celsius. Once ready, we add 0.6 milliliters of reagent 2 and cool for 2 minutes, 1000 RPM at 25 degrees Celsius, and we would have our sample ready for the PCR reaction. We can see clearly the Condagene extraction kit complex is straightforward and requires just a couple of reagents. And if this protocol has seemed simple to you, let's see how we can make it faster and more effortless with the Condagene quick extraction kit. We have basically the same protocol, which we start with a smaller elution sample, 0.3 milliliters instead of 1 milliliter. We centrifuge for 10 minutes, 13,000 RPM. We obtain our pellet and again discard the supernatant. We resuspend this pellet by adding 0.2 milliliters of the single reagent, homogenize well in a vortex for a few seconds, and take to a thermoblock for 5 minutes, 1000 RPM at 95 degrees Celsius. And just like that, in practically a single step, we have our sample again ready for the PCR reaction. Simple protocols, minimal manipulation. Now we go ahead with the PCR reaction using the Condagene Legionella kit. We have here our ready to go sample from the extraction step. We run the centrifuge for 1 minute at 13,000 RPM. This time we are interested in the supernatant. This is our sample, from which we take 5 microliters and add to a PCR tube or a well. To this PCR tube, we also add 4 microliters of our blue cap tube with the qPCR mixture that we have already ready. and also 11 microliters of the PCR grade water. So in our PCR tube, we should have 20 microliters in total, and that would be our sample tube. So how would we load a PCR plate? On one hand, we have our PCR tube or well with the sample, then another tube or well that will also contain the same amount of qPCR mixture and water, but 5 microliters of the positive control, the tube with the yellow cap instead of the sample. And then again, the same compounds, qPCR mixture and water, but instead of the sample, 5 microliters of water, that is more water for the negative control. With all these reagents loaded, we proceed to add our program in our equipment, every step with its temperature and time and number of cycles. Contagion Legionella is indicated for quantification, and to make the calibration curve, the positive control would be used. This procedure is fully indicated in the instructions for use. And these would be the possible results that we could obtain. So in the in the FAM channel, we would have the detection of Legionella, and in the X channel, the internal control. Is how each of the substances would be read. So if we have the FAM channel positive, we would be talking about a positive sample for Legionella. If FAM is negative and X is positive, the sample is negative. But for example, if we had this result, a negative PCR with a positive culture, we could be talking about possible contaminations or some problems that have occurred there's been an inhibition in the detection of Legionella, but that the PCR works well. And if both channels are negative, it means that some problem has occurred and the reaction is being inhibited. We have thoroughly reviewed Contagion solution for Legionella. So let's see how it can be used in conjunction with culture media in certain situations. For this, it is necessary to show that although culture media are indicated as the reference method, if not always, almost always, for ensuring water quality, some European regulations have begun to introduce this rapid technique, the PCR, to allow its use. This also comes in part from the experience of the application of these regulations, the surveillance of Legionella in recent years, the trends, availability of new techniques and innovations, and in general advances of technology that allow the improvement of these processes. One of these is Directive 2020-2184 of the European Parliament, and relative to the quality of water for consumption, where Legionella and the value that must be met are indicated, and for which the method of analysis is, of course, ISO 11731 method by microbiological culture. But very interestingly, ISO 12869 is also included as a complementary method, and as you can imagine, qPCR is specifically mentioned as an alternative method above others. And just to mention, at a national level, in Spain, the Royal Decree 487 from 2022 for the prevention and control of Legionellosis was recently updated, where the ISO 11731 culture method is also the reference method, but qPCR is mentioned again, in particular as a complementary method for the analysis of Legionella. And it is where Contagion has been specifically developed following this ISO 12869 for the detection of Legionella by qPCR, and where it can fit perfectly and following as well the recommendations of both regulations. So how could this be implanted together with microbiological culture, because it is yet not quite clear. As you can imagine, the regulations are just upon someone understanding and somebody else understanding something completely different. They do not specify the situations on where the qPCR, the ISO 12869, can be used. But for this, Paloma Belber from the Public Health Laboratory of Galicia from since some years ago has already been proposing an approach designed to improve the flow of Legionella analysis previous to these regulations updates. So in this case, specific, we can assume that we found a sample with a qPCR result higher than allowed. Paloma then proposes repeating the PCR to make sure that the result is accurate. As we can see, there can sometimes occur some problems, and then at the same time start culturing the sample for which we would already have it ready and treated, and if you will have this time reduced of the treatment of the sample. And then we would have 4 possible outcome scenarios. If both PCR and culture are negative, then there would be nothing else to do. These results, of course, would be taken as valid. If the culture is positive, regardless of the result of the PCR, then we would have to follow the process of samples confirmed by culture. It has to be done according to the legislation. However, if the culture is negative and the PCR is positive, further investigation would be required, since there may be certain reasons that, again, have interfered with the result. So you have to verify just a bit of what might have happened if you have a false PCR positive, or maybe some inhibition in the culture method. This has to be, analyzed. And a practical case on how to implement a PCR and a culture strategy in, for example, a health emergency, a sanitary emergency situation. We have, I don't know, in a public health laboratory, a sample has arrived and it is associated with a clinical case or an outbreak. We receive it, and then, of course, we have an urgency to obtain the results. If positive, that this is the case, we can begin our qPCR method. And do we get a positive PCR result? Then we must continue with a culture. Also, again, with the advantage that our initial sample is ready, and then we can continue with the extraction, and we can continue with the culture immediately. And we have already an early presumptive result. And if negative, the qPCR is negative, then we would just report the result. Because, again, a confirmation would not be necessary if this qPCR kit method that you are following has all these internal controls as Condagene kit. And in the case that it is not an urgent sample, we just continue calmly, well, calmly not, but more without the pressure to obtain a rapid result. We continue with our microbiological culture method, and that is it. And it is where Paloma Belber proposes qPCR as also a first sample screening, and that is very in line with the updates of the regulations that we have reviewed. It is complementary. It is not to be the reference method, but it is to be used in certain situations that a quick result is required. So in a laboratory with samples and real cases, what would this implanted technique look like? We can see clearly in this graph, we have on one side dark blue for the PCRs carried out, orange for the positive PCRs, and in yellow, the positive cultures that were made only from the positive PCRs in orange. And most important of all for us in this case would be the avoided cultures in a lighter blue. So we would have 2,272, 2,000 would be better, but 272, 121, 175, 150, almost 91, 161, all of these avoided cultures. And if these numbers, these 272 cultures, 161 cultures do not impress us, let's just think that for just a single sample, around 30 plates are required. All this in addition to the complexity of the culture method, and reminding that these media are very difficult to discard due to the antibiotics in their formulation. It is well established that a combined strategy of qPCR and culture media gives us a very high speed and confidence in obtaining the results. The regulations do not allow yet for the or completely or transparently use of the qPCR, but it is for the sector, for the laboratories to push this method, because otherwise we are gonna keep waiting for the regulations to keep up and say, yeah, the qPCR works. So it is, again, the job of the industry, the job of the people working in laboratories to push for this change, for this reform, because it has always been this way. Laboratories and the industry, microbiology, has always pushed using innovations, other methods, and then the regulations, the legislation catches up. So it is up to us to introduce this qPCR as a combining strategy. Obviously, never letting go of the culture media, that they are the reference. This method is the reference method for a reason. So just to keep in mind. And then just to finish, we're going to quickly recapitalize all the products that we have available in Conda Lab. We have, of course, our Conda gene line for the detection by qPCR. We have the 2 kits for extraction that we just saw, and for detection of Legionella, also Salmonella, although we have not reviewed it today. On the equipment side, we have our CDL 96 thermocycler, which is a very open and very versatile system. It has 4 reading channels that can just have a 6-channel option and would allow a multiplex. The software, I have used it. It is very intuitive and user-friendly. It has a lot of functionality, the software. It is obviously tested, optimized for the use with Conda gene kits, but as I say, it is not limited to. And well, if you are more interested in learning about the CDL 96 thermocycler, you can just let us know, and we will send you all the information about this equipment. And we wouldn't be culture media manufacturers without having all the media available. All the products for the protocol according to ISO 11731 are available with the same base. Depending on the added supplement, you can have BCYE agar, BCYE with antibiotics, BCYE without cysteine, GBPC, MWY. And of course, there has to be ready-to-use plates to avoid all this complexity to prepare the culture media. And these plates are very convenient to start right away your testing without any delay. Finally, just what are we working on? We have practically ready the Conda gene kit for Listeria monocytogenes, and in development, one of the reagents I mentioned for the viability PCR, with which the DNA of non-viable cells or free DNA is neutralized to avoid false positives, one of the limitations of the qPCR. And in some other R&D projects, we have kits for Listeria, CAMPI, STAC, and we have to do it for multiplex since we see it is an option for Condagene. Not least, mention all the resources you have available in Condagene in terms of technical documentation, in addition to technical and safety data sheets and analysis of certificates. We have lots of bibliographic material, kit verification reports with exclusivity tests, matrix of conclusions, etc., stability studies for transport at room temperature, and also for the freezing and thawing cycles we mentioned of the reagent, shelf life analysis, and of course, detailed instructions for use with the kit protocol, information on related standards of the equipment required, and the interpretation of results. We have finished the webinar. I hope you have found it interesting. Any doubt, of course, we will be happy to answer it, and we are at your disposal for any issue that may arise. I took a long time with the presentation, so just quickly a couple of questions I'm going to answer right now. Okay, we have a lot of questions. I appreciate the participation. Let's see. Okay. How many PCRs can I run with the amount of positive control? This is going to be determined by the needs of each laboratory. We cannot say just a number. That's why we set a range between 20 to 50. So if you follow the guideline of reconstitution of the positive control, 100 microliters, it will give you for 20 PCR. But if you do not quantify and you do not do the calibration line, that would be the case, for example, for Salmonella, that you are just looking for absence or presence, you can add up to 250 microliters in the positive control, and that would give you for 50 reactions. But that depends on your needs and what you are looking for, what you require. How, oh, I'm sorry, I'm having, how would I detect an inhibition in a PCR? Okay, so the case of a PCR inhibition, as we saw, the internal control is not being amplified, which could be happening. So the most easy thing to do, or the first thing to do, would be to make a 1 to 10 dilution of the sample material, because normally it can be inhibited by a high DNA load of the initial sample. So if it's diluted, the sample material with this, sorry, the PCR signal would be given without any problem. What matrices have you validated for Legionella? In this case, we have validated for drinking water, very much in line again with the regulations, Directive 2020-2084 of the European Parliament. Cooling towers, of course, and sanitary hot water. All of this information can be found in the validation reports. It is indicated. Combined strategy for Legionella positive sample, how many days it would take? I understand you're meaning a positive qPCR sample. So you have a presumptive result. So then you start with the culture. So you would go right ahead with the isolation, because you would already have your sample treated, because the treatment of the sample is the same for either qPCR and culture. So you are saving these days of the sample treatment, and you begin with the isolation, and then I think we're talking about 15 days. But this has to be done, of course, if you have a positive PCR sample, but then you can start to take steps. If it's, for example, a sanitary emergency, a health emergency, clinical case, you can say, hey, I think it might be Legionella. We are going to confirm it, but meanwhile, you can take steps. Investigate the source, investigate where the Legionella might come from, etc. You can begin the process. So it helps for this, even though, again, it's going to take the time of the culture that it's going to take. You cannot erase it. You cannot make it faster. The culture media method is going to take time, regardless of whatever you use. So we have a few other questions. So I'm gonna answer these by email these days. Do not worry, not one will be left unanswered. We want to invite you to our next webinar that will be in December, and we will talk about the analysis of non-sterile products in pharma. The communication, we will be sending it later. I think November. We hope this webinar has been interesting for you. We invite you again to subscribe to the newsletter and follow us on LinkedIn. We are very active, and through these channels, we communicate all the news updates we have in terms as well of products, not just webinars. And as you can see, we are very active with the Condagene line, and there are very interesting things coming, and also from other product lines, not only Condagene. So as always, you're going to have both the recording and the certificate of assistance. You will receive it tomorrow, more or less around this time, maybe an hour later. And as I said, we appreciate your assistance and participation, and we hope to see you at the next Conda Lab talk. Until next time.